everyone. I'm Rick Lifton, president of the Rockefeller University, and I'm delighted to welcome you to another of our sessions, Virtual Discussions with Genuine Experts. This is the second in our series of webinars about the pandemic and the work that Rockefeller scientists are doing to combat COVID-19. My thanks to everyone for a remarkable outpouring of generosity and support for the university at this challenging time. We're incredibly grateful. We have a remarkable group of scientists in 20 Rockefeller laboratories who have rapidly embarked on important work on COVID-19 in collaboration with colleagues around the world. Today's virtual discussion is with one of those scientists, Jean-Laurent Casanova, who is a pioneer in research on genetic predisposition to infectious diseases. Before we hear from Jean-Laurent, I will give you an update on some of what's happened over the last 2 weeks in the pandemic. So this is the coronavirus. As we've talked about before, the coronavirus is very small compared to a cell. Those pink stalks that you see are the spike glycoprotein that is used for the virus to gain entrance into cells, and it's a major target for therapeutic intervention, particularly for vaccines. So this is the status of the pandemic globally. This is from the Johns Hopkins website, and as you can see, as of this morning, there were 3.7 million cases around the world with over 260,000 deaths. In the United States, there are 1.2 million cases currently, and as you can see in the lower right from the trajectory of how cases have grown over time, it's still going up relatively linearly when you consider the entire world. However, the pandemic is not equally distributed across the geographic landscape. As you can see in this map of the United States, in the US, cases are highly concentrated in the Northeast and are very sparse in other parts of the country. So if you look to the right, you can see how this varies state by state. So for example, in New York, there are nearly 17,000 cases per million in the population of New York and 1,000 deaths per million in the population as well. But it's quite variable across other states. So while Michigan has 4,000 cases per million and 400 deaths per million, there are other states that have much lower numbers. For example, Montana has only 400 cases per million, so about 40-fold fewer cases per million than the US, and only 15 deaths per million, about 100-fold less than what we see in New York. And this is not simply because Montana is small and remote. Other large states, such as California, which started off at about the same time New York did with this epidemic, has far fewer cases per million and far fewer deaths per million. So there are many factors that contribute to this variation, and we don't have a complete understanding of what those factors are as yet. So we can see that in addition to regional variation, there is temporal variation in the shape of the epidemic. So on the left shows the slope of the accumulation of new cases on a daily basis in the New York metro area, and you can see that it went up to a peak around the end of March and has been steadily coming down. And in the right-hand panel, in contrast, you can see that for the rest of the United States, the slope has generally been going up. So we have within the same country two very different epidemics, where New York is past its peak and is going down, and the rest of the United States is continuing upward. And that's a combination of some states that are still going up and others that are now going down. This shows from one of Governor Cuomo's presentations yesterday how many new hospitalizations there are per day. So at the peak of the epidemic, there were over 3,000 new hospitalizations required per day for COVID-related diseases. And this has been coming down quite steadily due to the very active efforts at social distancing, hand washing, wearing masks, staying indoors, staying in isolation from one another with the strict stay-at-home policy with the exception of essential services. So that number has come down to, on May 5th, day before yesterday, rather than 3,000 new cases per day, we're down to 600 new hospitalizations per day. And that's dramatic advance. Next slide, please. This is paralleled by a dramatic reduction in the number of deaths attributed to COVID-19 every day. Back on April 14th, we're at near the peak with 750. The maximum number that we had recorded was 799. And in the last 3 days, we've been down around 220 to 230 deaths per day. Obviously, still many lives being lost on a regular basis, but not nearly as many as we had previously. While death is a lagging indicator because that obviously occurs late in the course of the disease, the new diagnosis of cases is more of a leading indicator. And the next slide shows how dramatically that has changed in Manhattan over the last month. So back on April 8th, the 3-day average number of the percent of of all tests that were positive, these are tests for the RNA of the virus. So this is a test for current infection, showed that nearly 50% of all tests were positive by this RNA test. And that has been dramatically coming down over the last month. So as of May 6th, the 3-day rolling average had come down from nearly 50% to under 10%. So this is dramatic progress in the accumulation, the rate of accumulation of new cases. As a consequence of testing being so sparse, it's important to say that most people who have the disease do not get tested, and consequently we have had little idea of whether we were just seeing the tip of the iceberg or there was in fact a mountain underneath what we're actually seeing with these RNA tests. And in just the last 2 weeks, we've started to get new information on this from the use of antibody tests. So antibody tests do not test directly the presence of the virus. They detect the presence of antibodies to the virus that your body makes when you have been infected for some period of time and your immune system can kick in to mount an immune response that helps defeat the virus. So these tests have come online in the last couple of weeks, and data shown here is from different regions of New York. And in the left panel is everything but Manhattan and Westchester. And you can see that the average, the percent of people who have positive tests for these antibodies is generally 1 to 3% of New York. So upstate New York, central New York, and many other places have between 1.2 and 3%. Western New York has 6%. But on the right, you see what you get when you test people in New York City overall. About 20% of people in New York City have antibodies to the virus, indicating that in contrast to the roughly 2% of people who are diagnosed by an RNA test, there in fact is a much bigger mountain underneath that in people who don't get tested. There are another 20%, or roughly 20% in total, who have been infected, telling us what we believed all along but didn't have direct evidence of, that most people who have the virus do not require hospitalization, do not present for testing, and are simply staying home and riding out whatever illness they have, including people who may be completely unaware that they have been infected. We don't know as yet what the shape of these curves is going to be. This is a slide that I showed the last time we met, which showed different alternative outcomes of infection in the population. In the orange curve, showing that we hit a peak, everything goes away, and we never see the virus again. That's increasingly unlikely to be true based on experience around the world, including in the United States. A second model is where we hit a peak and stay at a peak, and that's the yellow line, where we just progress on and never actually manage to bend the curve and get the number of cases to a low level. And the third possibility is the blue line, where we hit a peak, manage to contain further spread of the virus, and it diminishes to a low level. But once we start coming out of our isolation, we may go back to seeing cases recur again. The best guess is that we're somewhere between the yellow and the blue lines in how things are going. We clearly are doing a I think, an outstanding job in New York City in driving down the incidence of disease. You've seen the curves that there's dramatic reduction in the incidence of new cases as well as the number of adverse outcomes. And yet we don't know at present what will happen when we start coming out of our quarantine and sheltering in place. So one of the obvious challenges is what's going to happen as we try to reopen the economy in New York and elsewhere around the country. And we've talked last time about what the ideal conditions might look like and how this compares to reality. We would like to have very low levels of infection in the population and enough testing capability with rapid turnaround to allow quick identification and quarantine of all those who become infected. We do not have that capability today in New York or anywhere at the numbers required. We still barely have enough reagents to diagnose people who require hospitalization, much less ability to trace contacts of individuals and identify them and isolate them. So we have a number of challenges going forward. And it's clear that we are going to have to maintain social distancing, wear masks in public spaces, including at work, to reduce the risk of viral transmission to one another. And we will require exceptional efforts to keep our public spaces sanitized, especially public transit. Since our last discussion, I'm pleased to say that the governor has really bought into all of these concerns in a major way, and you're well familiar with his new moves to close down the MTA between 1 and 5 in the morning in order to allow sanitization of public transportation, because as we start moving millions of people a day on public transit, that's going to be a major threat as a source of recurrent infection. There are a number of important emerging clinical observations since our last meeting. The first of these, and perhaps most importantly, is the emerging role of vascular disease in this pandemic. It is now apparent that many patients experience large and small vessel thrombosis, the clotting of small blood vessels or of large vessels that affect not just the lung, but are found in many organs, including the brain, the kidney, and thromboses that occur in the veins of the leg that may migrate to the lung and cause pulmonary embolisms. Part of the hints of these that came online early were young adults who typically have very few manifestations of the disease, who are presenting with large vessel strokes, which are very uncommon in the general population, but started being seen in rare patients who have been infected with the virus. And just in the last week, we've heard about a new syndrome in children called Kawasaki disease. And Kawasaki disease is a rare disease of almost exclusively of children that typically involves inflammation of the blood vessels that can cause quite severe diseases of the heart, of the extremities, and so forth, that has been of mysterious origin, but has now been seen in quite a substantial small number overall, but it's such a rare disease to see it in dozens of children with COVID-19 infection has indicated that this is clearly related to this disorder. Whether it is virally mediated directly or whether it's related to the immune response to the virus is presently unknown. Other important questions are, we really don't know whether recovered patients are immune to reinfection, and if they are, how long will that immunity last? Coronaviruses are common cold viruses in many cases, and we typically do not make develop long-lasting immunity to coronaviruses. So we don't know if we had an effective vaccine, how long that would be effective. And for people who have recovered, we don't know whether they are in fact immune to reinfection. We suspect that because they recovered and they've have some antibodies, we know they've responded. We suspect that they will have some level of immunity, but we don't know what that level of immunity will be. We have learned in the last 2 weeks that patients who have recovered Some have neutralizing antibodies that are capable of preventing the virus from infecting cells in our body, but many do not, leaving open the question of whether antibodies are important for most patients who have recovered from the virus. We continue to have limitations on viral testing, which is a major barrier going forward. We need much better testing, much more rapid turnaround if, as we return to work, we're going to be able to maintain the safety of our workplaces. Importantly, this highly specific serologic testing is rapidly becoming available, unlike the RNA tests for the virus, which are hard to manufacture at scale. These tests for serology, for the antibodies to the virus, will be available in millions of tests. Roughly 100 million tests per month are likely to be available starting in about a week from now. So as we go forward, another important development when we talked last time was wondering about the prospects for vaccines, and there's been some dramatic progress in just the last 2 weeks on this. Several have already entered clinical trials, and these are both interesting vaccine candidates that are being prepared, taking quite different strategies. The two that are well into the clinic now in phase 1 trials, where they're basically testing safety, but testing safety in at-risk populations for exposure, so we may learn something about whether the vaccines have efficacy in the relatively near future. So one of these is a vaccine candidate from an academic group at Oxford, who have been interested in making vaccines for over 20 years, have a lot of experience, and had experience ready to go with an attenuated virus from monkeys. And this virus had been previously shown to be safe when given to humans in the context of other vaccine development trials that they had been working on. So they were able to use this vector for rapidly developing a trial candidate for the SARS-CoV-2 virus that causes COVID-19. And so they inserted the spike protein into this virus and have been injecting these into humans after showing some good evidence of safety in animals, including in macaque monkeys. So this is nice evidence in animal models, and this has led to the early entry into clinical trials in humans. And it's been estimated that by September, this could have sufficient signs of efficacy to go into at least phase 3 trials, if not beginning use in humans. They estimate that they can have 1 million doses ready to go by the end of the summer. But obviously that's a long way from scaling up from over 1 million to over 1 billion. So there's a long way to go, and it's still a ways in the future. A second vaccine candidate that shows some promise is one from Moderna, which is a company that has been pioneering the idea of making RNA-based vaccines, where instead of putting a protein into people, you're putting the RNA into people that will enter cells, make the encoded protein, for example, the spike protein of the SARS-CoV-2 virus, and we will then use that protein being made by our own cells to mount an immune response that may be more efficient and efficacious. So this will provide some evidence for demonstration of safety and efficacy. The timeline to widespread use is obviously an open question, whether these vaccines run into any challenges as they go through clinical development. And we will continue to watch this with great interest. So last time we had talked about therapies that were in common use, and we've seen some progress on a number of these fronts. Remdesivir has shown some evidence of efficacy that has led to an emergency use authorization from the FDA for patients with extreme cases of COVID-19. In the clinical trials, it reduced the time to recovery from 15 days to about 11 days, showed a trend that was not significant to reducing death. The data has not yet been fully released, but we already have emergency use authorization. Everyone looks forward to seeing the data on remdesivir under a magnifying glass rather than the summaries that are all that we've seen so far. Hydroxychloroquine, which has been widely used, has not come through. You hear less and less about it. It's mostly been used as compassionate use with very little organized randomized controlled trial activity. But we have heard very few reports suggesting that this is going to be useful in the clinic. And I think this is like the dog that hasn't barked over the last month from Sherlock Holmes' tale. Lopinavir and ritonavir were not terribly good candidates to begin with, and randomized controlled trials have shown that they don't work. Immune modulators have been widely used to try to prevent the progression of lung disease. And again, in the category of the dog that hasn't barked, the interleukin-6 antagonists are not leading to frequent reports of favorable response. We have not heard reports of readout of trials of randomized controlled trials as yet, but I think enthusiasm for these agents is diminishing. There are many other drugs that are being repurposed for trial. So these repurposed drugs come from large-scale screens for small molecules that inhibit viral replication in cells that have been safe in humans from prior clinical trials. About 100 candidates have shown some activity, and many of these are now in small clinical trials, including cysteine protease inhibitors, PIK5 inhibitors. This is an interesting drug that failed phase 3 trials for other uses, but it fails to acidify the endosome where the virus has to escape from. And this has shown a very potent activity in inhibiting infection by the SARS-CoV-2 virus in cells in vitro. And there's a significant amount of interest in this as a drug. Tamoxifen is a drug that shows weaker activity, but is one that is there's a lot of experience with in human trials. And referenced at the bottom right is a paper that reports the results of a large-scale screen for these repurposed candidates. Next. So at Rockefeller and elsewhere, there's a lot of activity trying to develop therapeutic or prophylactic antibodies in small molecules. Neutralizing antibodies, we talked about at the last session. Michel Nussenzweig is charging ahead with this effort. He's now screened more than 150 patients who have recovered from infection with the SARS-CoV-2 virus and has identified a number of antibodies that are potentially interesting for their ability to neutralize the virus. Targets, developing new medicines that might inhibit the virus. The RNA-dependent RNA polymerase, the helicase that unwinds double-stranded RNA to allow replication of the virus, and the proteases that are required to develop all of the proteins of the virus from a single large polyprotein are all essential proteins in the life cycle of the virus. And at Rockefeller, Seth Darst and Tarun Kapoor and Tom Tuschl and Charlie Rice have projects underway to try to identify novel inhibitors of these targets. So this gets us to novel therapeutic targets and assessment of risk coming from genomic and human genetic studies. Charlie Rice, next time we get together, we'll talk about CRISPR screens to identify viral and host genes that are essential for viral replication. And on the right-hand side is Jean-Laurent Casanova, who is going to speak to us further this afternoon. And so now I want to turn to my introduction to Jean-Laurent. Jean-Laurent Casanova has pioneered the understanding of rare mutations that produce extreme outcomes from infectious diseases, ranging from tuberculosis to influenza. His extraordinary research has achieved wide recognition. He's received the highest scientific award of the American Society of Clinical Investigation and the INSERM Grand Prize, which is the highest prize for biomedical research in France. He's also an elected member of the U.S. National Academy of Sciences and the National Academy of Medicine. When the COVID-19 pandemic began, Jean-Laurent immediately turned his attention to studying young patients with unexpectedly severe disease, and he has been systematically collecting and analyzing the genome sequences of DNA samples from these patients from around the world. His goal is to identify rare mutations in their DNA that render them particularly vulnerable to this virus. This work can identify basic mechanisms that normally prevent severe manifestations of infection, and these might be manipulated for benefit, not just in these rare individuals, but in other at-risk people as well. At the end of Jean-Laurent's presentation, we will answer your questions. Many of you have already submitted questions when you registered, but you're also welcome to submit questions now or anytime during the session using the Q&A function at the bottom of your screen. We will do our best to answer as many questions as we can before the end of the hour. And now it's my pleasure to turn over the podium to Jean-Laurent.
Thank you. So I am Jean-Laurent Casanova. I'm a pediatrician, and my work is at the intersection of 4 fields: medicine, genetics, infectious diseases, and immunology. Over the last 20 plus years, our lab has shown that life-threatening or lethal disease in the course of primary infection by one or another virus, bacterium, fungus, or parasite in previously healthy children, adolescents, or adults can be caused by genetic variations that in our jargon we call inborn errors of immunity, and that remain clinically silent until exposure to an infection by that particular microbe. And therefore, when the outbreak of COVID-19 occurred in February, we naturally decided to try to tackle this problem. And that's the story, the 2-month-old story, and the plans we have for the future that I'm going to share with you in the next 20-25 minutes. So we have decided to try to understand severe life-threatening COVID-19 in previously healthy children, adolescents, and also adults, young or middle-aged adults. As you will hear during my talk, we have not done that as a solitary exercise. We've actually launched an international consortium following the pandemic in a way. And in the final slides of my talk, I will tell you more about this network of pediatricians and internists worldwide that work with us. So you are familiar with the idea that life-threatening COVID-19 can strike previously healthy people. It has been covered in the press. That's a paper in the Washington Post a month ago that drew the attention of the public to this problem. Until the beginning of April, the common belief was that severe COVID would strike mostly, if not exclusively, the elderly population. And unfortunately, young or older individuals with comorbidities, that is, with an underlying medical condition. But it gradually appeared that severe COVID can strike previously healthy people. And that is really the problem that we're trying to tackle. Now, before discussing this particular problem and the hypothesis that we test and how we test it, let's take a step back and review together what is life-threatening COVID-19. So it comes in multiple flavors, but I've shown you here 2 images of pneumonia or pneumonitis. These are the upper 2 panels. And you have in the bottom panel encephalitis, that is an infection of the brain. And also, as alluded to earlier by Rick, a vasculitis. This is a heart ultrasound that was taken a couple of days ago of a child with Kawasaki disease. And what you see here is, if I can show, what you see here, that's a dilated coronary artery. So life-threatening COVID comes in multiple ways. And the important information that, again, Rick alluded to earlier, is that these severe forms of COVID are actually a minority if you take into account the global pool of people who've been infected. On the left-hand side here, you see that only the red circle, these are the severe cases. The symptomatic cases are the dark gray, and you have a much larger group of people infected who do not develop clinical manifestations. On the right-hand side of the slide here, I want to try to help you put COVID-19 in the context of other infectious diseases. I have plotted here on the x-axis the risk of life-threatening disease among infected individuals, and on the y-axis the number of different infectious agents that can infect humankind. And I've estimated, this is only an estimate, that probably around 1 in 1,000 infected individuals, roughly, again, this is an estimate, can develop life-threatening COVID. This is more severe, the proportion is higher than, say, with seasonal influenza, that is life-threatening in only 1 in 10,000 people, roughly. And COVID-19 in that regard is comparable with the 1918 Spanish influenza, the 1918 Spanish flu. So the message here is that there is enormous inter-individual clinical variability in the course of infection by coronavirus. This is a general observation for infectious diseases, and it's been known for a long time. It's been discovered at the turn of the 20th century, and perhaps the paper that best posed the problem and came up with ideas, solutions to this problem is a paper by a former Rockefeller faculty, René Dubos, who published in 1955 in the Scientific American a beautiful paper with this provocative and eloquent title, Second Thoughts on the Germ Theory. And the abstract is clear and beautiful. Everyone harbors disease germs, yet not everyone is sick, suggesting that germs are less important in disease than other factors affecting the condition of the host. So this is really a beautiful paper that I encourage you, all of you, to read. And this COVID-19 pandemic in 2 months has already provided us with the possibility to identify 2 major risk factors that are shown here: age and ailment, that is an underlying chronic illness. I have plotted on the x-axis the decades of life from birth to over 80 years. And on the y-axis, again, the risk of death per infection. Please pay attention to the fact that on the y-axis is not a linear scale. This is a log scale. There's a factor 10 between 2 little horizontal bars. So what you see here is strikingly that COVID-19 is much more severe, or much more often severe, in human beings older than 50 or 60 years of age. In addition, the presence of 1, 2, or 3 comorbidities increases the risk dramatically. If you are older, say, than 80 or 85 years, and you have 1 or 2 comorbidities, the risk of life-threatening disease can be higher than 10%. Conversely, if you're younger than 50 years, and if you do not have any comorbidity, the risk is much lower, less than 1 in 1,000, perhaps closer to 1 in 10,000. So the population of patients with severe COVID-19 that we are interested in are the people younger than 50 years without comorbidity. It is enigmatic. Why would these people suffer from severe COVID-19? And that's the problem that we try to tackle. I've represented here in another way the risk of severe COVID as a function of age and comorbidities. The older you are and the more comorbidities you have, the higher the risk. That's why there are more little red symbols here on the upper and right part of the graph. Now, there are a few individuals, young, without comorbidity, who develop rare idiopathic severe disease. Idiopathic is a medical term that essentially means unexplained. So what we hypothesize is that these patients are sick against all odds in a way because they carry germline genetic variations, what we refer to as single gene inborn errors of immunity. Single gene because we think there's 1 gene mutated per patient, and inborn errors of immunity to stress the idea that these deficiencies are present at birth. A second group of outliers that we want to study in a second step genetically are conversely people who are older and who may have comorbidities and who surprisingly not only do not develop disease upon infection, but also for some of them are not even infected. And these people resistant to infection, we think, may also perhaps carry single gene variations in their genome. Now, this idea did not come out of the blue. There was no epiphany. This idea essentially follows 35 years of research in the field of human inborn errors of immunity. And in the next 4 slides, I'm going to take you a little bit into the details of the field so that you better understand the approach that we've taken and that I will describe to conclude the talk. So from 1985 onwards, a number of teams described the molecular basis of what was then referred to as primary immunodeficiencies. I will show you pedigrees like this one on these 4 slides. The squares indicate men or boys, and the circles, women or girls. And black circles or squares indicate patients. The disease that I consider in these 4 slides is tuberculosis, an infection caused by a bacterium. Now, what you see here are 2 siblings, a brother and a sister, who developed TB. TB stands for tuberculosis, because they carry 2 mutations of the same gene, mutations that actually make them vulnerable to a wide range of infectious diseases, not only TB. They develop TB plus viral, fungal, parasitic infections. These are primary immunodeficiencies that profoundly disrupt immunity of affected children. These disorders were known from 1985 onward. When I started my lab in the early 90s, we discovered and then characterized a number of familial infections. That is, a brother and a sister, in that case, selectively vulnerable to tuberculosis, to TB. And we've shown that the brother and the sister can be homozygous for a mutation that remains silent until they're infected by one microorganism, the agent of TB. Now, what you see here is that the infection really runs in the family. It's a familial Mendelian genetic disease. In a third step, from 2007 onward, we've shown that not only familial infections, but also sporadic infections, that is, diseases striking one individual, none of their relatives being ill, could also be caused by these single gene inborn errors of immunity, because these defects, as we've shown, have incomplete penetrance. That is, in this family tree, you see there's a vertical bar for the sister indicating that she carries the at-risk mutation, the at-risk genotype, but she's not ill. That is, the penetrance is incomplete. And from 2007 onward, we've shown rare mutations underlying tuberculosis, but from 2018 onwards, we've even identified common genotypes, common inborn errors of immunity underlying tuberculosis. So the field, as you've seen, has evolved into 4 steps from 1985 to 2018. But these studies on TB and other infectious agents, as we'll discuss in a second, have provided proof of principle that life-threatening disease in the course of primary infection can be genetic and can even be monogenic, caused by single gene inborn errors of immunity. But tuberculosis is far from COVID. It's a bacterium. And the question you rightly ask is whether there are examples of viral illnesses of the lungs or of the brain or of the vessels resembling severe COVID-19 that can be genetic. And the answer is yes. Over the last 10 years, as shown on the slide here, a number of inborn errors of immunity have been shown to underlie selectively, exquisitely, and really specifically viral pneumonitis caused by the influenza virus, severe flu, if you like, also rhinovirus pneumonitis, Rhinovirus is an agent of common cold, and in rare patients, it can cause pneumonia. Helen Su at the NIH has discovered mutations in a gene underlying rhinoviral pneumonia. So that's for the lungs, but we've also discovered a set of mutations underlying viral encephalitis of the forebrain or of the brainstem. And at the bottom of the slide, I want to stress that in addition to these single gene inborn errors of immunity underlying severe disease among infected individuals, there are also known examples of single gene variations that make individuals resistant to a viral infection. A good example with which many of you are familiar are the variations in CCR5, which when carried in homozygosity, confer resistance to the human immunodeficiency virus. Now, if we analyze these genetic data from an immunological and viral angle, as shown on that slide, you have on the left-hand side a pulmonary epithelial cell and a neuron from the brain. And I've shown in red the gene products that are mutated in patients carrying inborn errors of immunity underlying these two types of infection. Now, what is interesting is that in both cases, for these two types of viral illnesses, the mutations that we and others have discovered disrupt cell intrinsic immunity. That is, they disrupt the capacity of cells that normally reside in these organs to control properly the virus. These mutations do not actually impair the leukocytes, that is the immune system in a traditional sense of the term. On the right-hand side, you have an illustration of CCR5 mutations conferring resistance. The CD4 T cells of patients homozygous for these variations cannot be infected by the human immunodeficiency virus. So this is to say that there are examples of monogenic viral illnesses that resemble severe COVID. So with this in mind, we decided in February to try to tackle severe COVID in young, previously healthy patients. And we launched a consortium designated as the COVID Human Genetic Effort. You have its logo on the upper left part of the slide. So what do we do? First of all, we recruit these patients. The recruitment essentially consists in asking patients younger than 50 years admitted to an intensive care unit with pneumonia or encephalitis without comorbidities to give us a blood sample. Then we sequence the genome, or what we call the exome. The exome is essentially the genes that are present in the genome. Then in the third step, we analyze these genomes. We analyze these genomes from two angles, what we call in our jargon genetic heterogeneity. That is, we search in patients one by one variations that we think are candidate variations. Another angle is what we call genetic homogeneity. That is, we search for variations that may be found in two or more patients. For example, a patient in New York may carry a variation in the gene, the same gene that a patient in Tokyo and a patient in Sydney. And then in the fourth step, we try to we test the hypothesis that these candidate variants are more than candidates and that they're actually causal, that they are responsible with the virus, of course, for severe COVID. That's the global strategy that we've adopted and implemented. So it has worked well. Over the last 2 months, we've already enrolled 500 hospitals worldwide in more than 40 countries. I have launched this initiative with Helen Su, a friend and colleague from the NIAID at the NIH. And in red here are the countries that are already involved. And we hope in the coming weeks and months to have more hospitals, more academic institutions, and more patients. Why do we need more patients? Well, because to detect genetic homogeneity, we need the greatest number of patients possible. It may be, again, that there's a patient in New York and we would never discover that the mutation is causal if we don't sequence a patient, say, in Singapore, because the second patient on Earth with that particular mutation would be in Singapore. It is therefore very important to enroll as many patients as possible. Once the patients are enrolled, we have their blood samples shipped to sequencing hubs. That is, we already have over 30 sequencing hubs. They're represented here. We have hubs in Japan, in China, in Singapore, in Australia, in the Middle East, in Iran, Saudi Arabia, Israel, also in Greece, Western, Northern, Eastern Europe, in Latin America, and of course, in North America, including Canada and the US. These sequencing hubs receive the blood samples, sequence the genomes, and then we analyze the data together. Now, we're currently analyzing the data. Our own lab has already sequenced 150 patients, and Helen Su at the NIH has already sequenced also, I think, over than 100 patients. So we're currently in the phase of analyzing all this data. And, I can't disclose anything, it's preliminary, but let's say that I'm optimistic. I think we're gonna find interesting variants. Now, what if severe COVID-19 were genetic in young and previously healthy patients? Well, it has implications. For the genetic cases and their families, first of all, we can provide genetic diagnosis, even genetic counseling. If there is a brother or sister carrying the at-risk genotype and who has not been infected yet, we can even think of a way to prevent or treat disease, or treat disease in these families. An example is, again, tuberculosis. Over the last 25 years, we've shown that gamma interferon is a cytokine that is critical for immunity against the agent of tuberculosis, and that the genetic disorders that underlie tuberculosis, in fact, are associated with diminished production of gamma interferon. Therefore, giving the cytokine as a medicine is efficient to prevent or treat microbial disease. Now, the hypothesis here is perhaps that the genetic mutations or variations that we find disrupt a pathway, an immunological pathway with which we're familiar and for which there is already a medicine. A good example or good news would be if we found mutations that impair the production of other interferons, the interferons alpha beta, which are antiviral. Because again, this is another medicine that you can find at CVS. So that is the set of implications for genetic families. Now, it may also have implications for the other cases, including other patients or patients with or without comorbidities. Because it is possible that even though severe COVID can occur, say, in someone age 85 with two comorbidities, not because of a monogenic variations, as we've just discussed, it may still be the case that the mechanism of disease that is altered, say, by age or the comorbidities is similar, if not identical, to the mechanism of disease that is disrupted by the mutation in the young genetic cases. And therefore, the implications in terms of prevention or treatment of disease could be similar in these patients. Now, if you want to know more about this effort, I've indicated here our website, and I want to close this talk by thanking the team and the extraordinary institutions that have supported our effort over the last 25 years, and in particular, Rick, over the last 3 years, and in particular, in the last 2 months for his support of our effort. Thank you for your attention.
Thank you so much, Jean-Laurent. That was terrific. Jean-Laurent, you've talked about rare mutations with large effect. There may also be common variants with small effect, and I wonder how you think about those in terms of being able to predict who might be at risk.
Right, so we are also searching for common variants that have a large impact and common variants, the impact of which is more modest, as you say. To detect them, we'll need, probably 1,000 or 2,000 patients. So for now, we focus on rare variants as candidate variants.
Thank you. So there's an important question about anticipating the reopening of schools. Is there research on the contagiousness of kids? This obviously is a really important question as we look forward to reopening of schools in the fall. We know that children are typically not severely affected, but we do not know whether they are infectious, how infectious they might be, and whether the arc of transmission is most frequently from parents to children or from children to parents. This is obviously a critical question as we think about having children go back to school and whether they will be vectors for spreading the virus throughout communities. And this is a key research question that we need to get much better epidemiology on and is a key question for the future. There were a number of questions about antibody testing that basically revolved around how reliable is antibody testing and why is it so difficult? The simple answer is it's not very difficult. It's a very straightforward test. All you need to do is put protein on a plate and in a little well in a microtiter dish and add blood and see whether it reacts or not. The problem is that many of the tests were rushed into production, and the FDA allowed many tests that had poor specificity to enter the market. So as a consequence, there have also been many tests that have not been licensed that are in the market as well. The FDA has started to crack down and only allow tests that have no more than a 5% false positive rate. But you can tell that if you had a 5% false positive rate and the prevalence of disease in the population was 5%, it would be a coin flip as to whether a positive test meant that you had previously been infected or whether the test was false positive. That would not be very useful to you in knowing whether you had previously been infected or not. The other point that is worth noting is, as I mentioned, we don't know to what extent having antibodies means that you will be immune to subsequent infection in the near term or in the long term. And this will require much more experience to gain more confidence in whether that's feasible or not. For you, Jean-Laurent, another question do you have a sense of what you think the biology will likely be underlying mutations? Will they be in cells that prevent them from being severely affected either by infection or progression, or will they be outside the cells that get infected from defects in the immune system?
Well, by analogy with the other viral illnesses we've studied, I suspect we find variations that affect pulmonary epithelial cells, or cells in other organs, as opposed to leukocytes. And I would therefore, favor the hypothesis that cells that are infected by the virus carry the variations that matter.
There's a question that has come in from your experience looking at the disease around the world. Do you think the environment has a role in susceptibility to infection and morbid outcome? Why do you think some countries like India that have high population density thus far has a lower infection level compared to places like Italy, Spain, and the United States?
Well, I'm not sure we have data that are compelling. It is difficult, I think, to compare India and Spain. The tests are different, the number of people tested, and so on and so forth. I think we have to be very prudent.
Jean-Laurent, in addition to variations in our genomes, how about variation in the viral genome? And does this explain some of the variation, and will mutations in the viral genome allow it to escape vaccines and require future development of yet additional vaccines or therapeutics?
That's a question for Charlie Rice in 2 weeks. I pass.
I think it's fair to say that the evidence thus far has not suggested a rapid evolution of the viral genome. And in fact, on the RNA polymerase, which is a major drug target, selection for resistance in the laboratory has shown that the virus doesn't do very well with the resistance mutations. So it might be harder for this virus to adapt to effective drugs than we might have been concerned with. Let's see. Jean-Laurent, with things like Kawasaki disease, do you think of the Kawasaki disease as being a primary consequence of the virus, or do you think it's likely to be related to the immune response to the virus?
So yeah, that's another great question. So you won't believe this, but I've been studying Kawasaki for 10 years. And the reason I've been studying Kawasaki is because 12 years ago, I reported two young children with primary infection by cytomegalovirus who developed Kawasaki disease in the course of primary infection by this virus. So I became convinced that it had a viral origin. And we've already sequenced the genome of maybe 120 patients with Kawasaki. And so far, we hadn't found any hit. But now that we have the COVID wave of Kawasaki patients caused by one particular virus, perhaps we're in the better position to crack this problem.
Do you think, from your experience with other viruses, do you think there are common pathways that are likely to underlie many host mutations that are causing severe viral infections?
Yeah, there can be an overlap. We already know there is an overlap because mutations in the toll-like receptor 3 pathway can underlie herpes simplex virus encephalitis and influenza virus pneumonitis. So I wouldn't be surprised to, discover overlapping sets of mutations.
Several people have written in inquiring whether BCG might be a factor in explaining why countries that use a lot of BCG seem to have lower rate of severe infections.
So yes, again, I'm not sure the data are compelling, and I'm agnostic. I'm willing to consider really almost heretical observations, but I would be very surprised if BCG protected from severe COVID.
So how would you go from finding a rare mutation that causes a severe form of infection to using that information to developing a new approach to therapy or prevention?
Well, if the mutation disrupts a known circuit, then of course it's easier. If the mutation is on a novel gene defining possibly a novel circuit, well then evidently there will be years of work to go from the mutation to a therapeutic success.
Is there any evidence that COVID-19 emerged from a laboratory by manipulation of the viral sequence?
Well, that's a question for Charlie Rice again. I pass.
I think the evidence thus far is strongly argues no. The mutations that distinguish SARS-CoV-2 from other viruses are not a small number of changes that you might anticipate would be found if a virus that pre-existed in the population had been taken out and intentionally manipulated and then released back into the population. On the other hand, if a virus had been taken into the laboratory for study and then got out entirely on its own without any intentional manipulation, but had this severe effect, you would never be able to tell with any certainty, but it wouldn't look very different from what had been a natural occurring virus in the population anyhow. So I think it's hard to distinguish those alternatives. There's a question, can CRISPR-Cas9 gene editing techniques be used against the virus?
Well, that I don't know, but it certainly can be used to try to test whether the candidate mutations we find are responsible for COVID, and we use that technique daily in the laboratory.
I will use that question to comment on the testing issue, which is an important one. The methods that we're using for testing for the virus are rather cumbersome methods that are not well suited to point of care tests or rapid turnaround in general. And the rapid turnaround instruments that we have are very low throughput. There are several ideas floating around in development that might provide larger scale capabilities, more rapid turnaround tests. And these largely revolve around a couple of technologies. One of them is technology that's found in every major research university, which is high throughput DNA sequencing. So the instruments that are providing these high throughput tests today can process about 3,000 samples per day, whereas the DNA sequencing instruments that are present in every research university in the country and in many biotechnology labs around the country can produce hundreds of thousands of tests in a single run. There are some technical aspects of how do you get these, the front end of the specimen collection and processing onto these instruments, but we have not been terribly imaginative in matching the scale of how we're doing testing to the challenge that we have of needing many more tests than we're currently producing. CRISPR technology offers another possibility for developing higher throughput tests that would be able to scale much better. There's a form of CRISPR in which recognition of a particular RNA molecule can trigger the Cas13 enzyme, which degrades all of the RNA in the vicinity of the enzyme. And that can be used to provide a signal that would indicate that the SARS-CoV-2 viral RNA is present in an RNA sample. And this is but one of a number of tests that are under development to try to come up with something that would be much higher throughput than what we have now. Another question about the virus, are there novel strains of the coronavirus that have become dominant worldwide and appear to be more contagious? I think the evidence for that is not at hand as yet. It's unclear that there's significant variation in the virus. Jean-Laurent, do you want to comment on that?
No, I really don't know.
Okay, so I'll pin you down on something that you might know something about, which is what do you think about the male-female difference in the prevalence?
Well, that is very intriguing, and an obvious hypothesis is that the receptor for the virus is encoded by a gene located on the X chromosome, and therefore variations of that gene or variations that regulate the expression of that gene may explain a greater vulnerability of men when compared with women. And that is one of the genetic hypotheses that we're testing in the lab.
And of course, it doesn't escape one's attention that the receptor for SARS-CoV-2 is the gene ACE2, which happens to be on the X chromosome. So that's an interesting idea. There's a question about response to vaccines, and it's been noted that the dengue fever vaccine ran into problems late after clinical testing because some children who were immunized with the dengue vaccine, when they then encountered dengue virus, developed severe consequences. Concerns about this with the SARS-CoV-2 vaccine?
Well, I don't know. I think that's the phenomenon known as enhancement. Is the idea also is dengue itself, which is often more severe during the second and the primary infection. I'm not aware of anything similar with the other coronaviruses, but I don't think we can exclude this possibility, even though it is remote but finite.
Several people have written in inquiring about COVID toe and whether that is a manifestation of vascular problem like with Kawasaki disease.
Right, and that is also seen in a group of disorders known as the type 1 interferonopathies that are conditions characterized by an enhanced production of antiviral interferons without a viral trigger. So I think these lesions probably attest that these kids produce high amounts of interferon to try to combat the virus.
Is it known whether COVID-19 alters a patient's immune system directly to make a patient more susceptible to certain other infectious diseases?
I don't think we have enough data to compare the rate of superinfection of the coronavirus pneumonia when compared with other viral pneumonia. But this is an important topic an important epidemiological and medical topic for sure.
Well, I see that the hour is late, and I think we better draw the discussion to a close now. I'm grateful to all of you who have stayed online beyond our allotted hour. Thank you very much for your participation. I want to thank Jean Laurent for a wonderful overview of his work and brilliant discussion. As you've heard, he's made remarkable strides in a very short period of time. We still have a lot of work ahead of us, and philanthropic support remains vital to our progress. Please see the next slide for more information about how you can contribute and help us combat the COVID-19 pandemic. I want to again thank all of you for joining us today. Our next virtual discussions webinar will be with the virologist and Lasker Award-winning scientist Charles Rice on Thursday, May 21 at 4 p.m. Charlie's pioneering work contributed to the development of medications that proved to be the cure for hepatitis C. He is now concentrating his efforts on discovering drugs that will help patients during the present pandemic while developing therapeutics to prepare us for future coronavirus outbreaks. I hope you'll join us on May 21st when he will speak about the innovative strategies he's using to discover drugs that are effective against COVID-19. Until then, all of us here at Rockefeller hope that you will stay safe and be healthy. Thank you very much for participating this afternoon. Good evening.